Watch it.
Sorry. The
Shoot, my bad.
Sorry.
Help you up.
Thanks. I'm sorry. I'm legally blind.
No, you're not. You're looking right at me, bro.
You couldn't tell by the cold open, this one is going to be a bit more of a serious video than last time. Calm down, you get your joke fixed soon enough in another video. But this one's about a topic that's pretty important to me. We'll get to why in a bit. Today we're going to be talking about a relatively rare eye disease called retinitis pigmentosa, or RP. As the name implies, it directly affects the retinas, eventually resulting in a tunnel vision that somewhat resembles what you saw at the start of this video. Together we're going to explore RP and how it affects the eyes. Then we're going to look at some relevant research, but as we take a deep dive into the literature, we're also going to explore the kinds of questions that serve as the foundation to neuroscience research. Now, without further ado, first, let me take you on a journey through the visual pathway. When light meets the eye, it passes through the cornea and the lens, both of which serve to focus the light before it lands on the retina. The retina itself acts kind of like a camera sensor, but it's not actually spread out evenly like a camera sensor. There's a central area called the fovea where light is focused onto. This part is good at seeing things with high definition, or acuity, and it's good at seeing things with lots of light and lots of color. The farther you get from the fovea, the lower your acuity is, but this area is better at seeing in the dark. The reason different parts of the retina are better equipped at seeing in different situations is all thanks to those good old rods and cones you might have heard of. Rods aren't really used in color perception, but they're pretty good at seeing in dim environments, which we call scotopic vision. Cones are only really useful in well-lit environments, or photopic conditions, but we can use them to perceive color. How we do that is a topic for another video, but to simplify, we have three types of cones, one for red, green, and blue light. Kind of like the pixels on a camera sensor. What really matters here though is the concentration of these rods and cones, which we call photoreceptors, throughout the retina. The fovea I mentioned a second ago contains loads of cones and pretty much no rods. The sheer density of cones in the fovea is why we have such good color vision there. And it's one of the reasons why our acuity is so high in the center of our vision where we need it most. Our peripheral vision farther out from the center still has cones, about 99% of them actually, but unlike the fovea, it also has rods. Now once light hits the rods and cones, this energy is converted into electrical signals, which then travel up the optic nerve to a structure with a long name that we're just going to call the LGN. This serves as a sort of relay that receives input from the eyes, and then directs it up to the visual cortex of the brain where it gets put together, little by little, until we have a full visual perception built. So where does RP fall into all of this? Well, most of it happens in the eyes as you'd expect. RP is a gradual process that actually starts with night blindness. In other words, one of the first symptoms is slower adaptation to scotopic conditions. And what do we know about scotopic vision? That's the rod's responsibility. As such, it's not surprising that this is exactly where the problem begins. First, rods begin to die in a process gradual enough that it can actually take years to realize that something's even wrong. Given that there are more rods in our periphery, this loss is more rapid on the outside, giving us that characteristic tunnel vision as the retina degrades from the outside in. But rods aren't the only cells that die though. Once most of the rods have gone, the cones begin to follow as well. This means that unfortunately, the degradation can reach the fovea, though the rate and totality of this do depend on the exact cause of the RP. More on that in a second. Interestingly, we don't actually know exactly why the cones are also affected, but one potential clue is the fact that the cones stay relatively intact until most of the rods are dead. You know what, let's see if you can figure out the prevailing theory on this. Post your guess in the comments and I'll let you know if you're right. Just keep in mind that we don't know that this theory is true, just that it's most likely based on what we know so far. Now, one last thing to note about RP is that it doesn't have one singular cause. It's actually a genetic disorder resulting from mutations in any of the 200 or so genes that have been linked to it. Because of this, there are many mechanisms that can lead from genetic bloop to cell death. The specific mutation that Sally has might make her lose all of her vision by the age of 50. Whereas Henry's mutation might allow him to hold on to that last little spot indefinitely. Just keep that in mind as we continue through this video, because it means that we're going to have to do a little bit of generalization here. But at the end of the day, each person goes through their own tunnel. So now for the fun part, the research. Remember at the start of the video when I mentioned the foundations of neuroscience research? Well, let's say you have a question about a neuroscience concept, like how do our eyes know how far to focus? In order to begin answering that question, you need to find something you can manipulate and something you can measure. Then you need to look at their relationship in order to understand the underlying mechanism. In neuroscience, there are three main relationships, seen here in the visual I flashed earlier. First on the list are stimulus-behavior relationships. In these experiments, the stimulus, which can be any kind of environmental energy like light, is changed or manipulated. Then behavioral responses to the stimuli are observed and recorded. So how about we take a look at a couple of examples of stimulus behavior experiments, starting with one conducted by Francia and colleagues where they tested the effectiveness of a nanoparticle treatment injected just under the retina. In a previous study, they demonstrated that these nanoparticles can actually restore visual activity in rats with essentially the rat version of RP. This study is just validating those results by demonstrating that brain activity and light-driven behaviors are also restored. They did this with multiple experiments, but we'll just talk about one for now. So in order to test light-driven behavior, we need a stimulus and a behavior. The stimulus in this case is light, obviously, and the behavior is this aversion to light that rats have that make them want to go hide in dark areas to avoid predators. In the lab, they tested this by just shining a dim light outside of a dark box and seeing how long it took the rats to jump in. They tested two types of nanoparticles and found that one of them actually restored this light escaping behavior to the level of healthy rats, suggesting that these particles can potentially rescue light perception to some extent. Another study conducted by Yamamoto and colleagues aimed to see whether certain metrics of visual function could be used to assess RP patients with ultra-low vision. One of the tests they used is called a full field stimulus threshold test, or FST test. This involves the use of a machine like this one, and actually this is the exact model used in the study, which shines a light of a specified intensity and color across the entire visual field, which is what we call the field of view. It did this by shining different intensities of red, green, and blue light to assess the cones, and white light to assess everything overall. So light is a stimulus again, no surprise, and the behavior in this case is simply the participant pressing one button if they can see the light and another one if they can't. Using this very simple design, the researchers found that participants with RP were more sensitive to blue light than they were to green or white light, and especially more than they were to red light. In addition to this, they took guessing into account and were able to determine that in order to get accurate results from participants with ultra-low vision, whose responses can be pretty inconsistent due to their extreme difficulty seeing, you need to consider the randomness introduced by these guesses. And now we move on to the stimulus physiology relationship. We already know what stimuli are, so what does the physiology part refer to? Well, when we measure physiological responses to stimuli as opposed to behavioral responses, we're measuring physical activity in the body. This includes things like heart rate, brain scans, or as is the case in a lot of neuroscience experiments, the electrical signals traveling through the nerves and neurons. One example of this can be seen in a study by Ahn and colleagues, where they were testing a different method of using retinal prosthetics in order to restore vision in integrated retina. The way these prosthetics work is actually pretty neat. See, while the rods and cones do die in RP, there are a few neurons in between these cells and the optic nerve. We'll focus on these just to keep things simple, which we'll call RGCs. These are mainly there to allow us to begin processing this visual signal right from the get-go, and also just to transmit the signal from retina to optic nerve. These RGCs actually remain intact in most cases of RP, which allows us to electrically stimulate them ourselves with prosthetics, which rods and cones can't do anymore. The issue with this is that the longer RGCs go without receiving stimulation, the more jumbled up their connections can get. In late-stage RP patients who get prosthetics, this results in their gain in visual acuity not being high enough resolution in order to escape being legally blind. In an effort to improve upon the prosthetics, the researchers in the study tried altering the pattern of electrical stimulation that they were using. The physiological responses that they measured were the electrical signals sent out by the RGCs, which they measured using an array of electrodes, kind of like the one you see here. What matters here is that the traditional pattern of stimulation led to loss in acuity, whereas their new pattern actually preserved acuity pretty well, which is pretty nice. A separate study by Stutzky and colleagues also looked at these prosthetics. However, they were more interested in documenting how RGC connections change in those with RP and to what extent. The researchers used a similar approach here, where the stimulus was electrical and came from the prosthetics, while the physiological response they recorded using another array of electrodes. This time, however, they compared RGC responses in mouse retinas with and without RP to see if there were any notable changes in connectivity. One thing they noticed is that there was statistically no significant difference in receptive field size. Receptive fields are just the region in which a stimulus causes neurons to fire. This, alongside the other data the researchers obtained, allowed them to come to the conclusion that these underlying RGC networks are preserved to some extent, which just shows how promising this hidden circuitry might be in the search for better RP treatments. Now lastly, we have a bit of a trickier relationship to explain, at least in my opinion. The other two we talked about both had stimuli in common. You'd present a stimulus of some kind to a participant, be it human or otherwise, and you get responses from that. Well, now we're going to take our focus away from the stimulus, even though it still is usually involved in the experiment in some way. What we'll do now is simply present the stimulus and measure both physiological and behavioral responses to see how they compare. This can be done within the same experiment, such as by having a participant answer questions about a stimulus while getting a brain scan, but it can also be done through multiple experiments within the same study. In the first study we looked at by Francia and colleagues, they performed multiple experiments too to assess whether those nanoparticles could get past the RGC issue. Some were stimulus behavior experiments, like the one I mentioned, and others were stimulus physiology, like this one that measured the pupillary light reflex, which refers to the physical changes in pupil size in response to changes in light intensity. And the way they did this is pretty straightforward actually. All they did was adapt the mice to the dark, shine a green light in the eyes for a few seconds, and then take a video of the pupil after they removed that light. With this video, they were able to determine a lot of different things, from the size of the pupil at different moments to the speed at which they opened and closed. The researchers ended up finding out that the particles they injected did restore this reflex in mice with RP. But I don't want to look at just this measurement. I want to look at the bigger picture like the authors did. When we do that, we see many different behavioral and physiological responses. Hiding in the dark, pupillary light reflex, and these are all influenced by the presence or absence of these nanoparticles. When we combine all of these responses, we can see the effectiveness of this treatment because of its close ties to the physiological and behavioral responses to light that we use to measure it.
Me gustaría recordar que es ver completo sin tener que buscar específicamente lo que voy a mirar, pero he aprendido.
Now you may remember at the very start of this video, I said that this topic is pretty important to me. You may have also been wondering who that woman was who was on screen for a bit. That woman is Norma A. Gomez. Her friends call her Angelica or Angie, but I prefer to call her Mom. At the time of recording this video, her visual field is restricted so much that she can only see one eye at a time when talking to somebody in conversation distance. That clip came from an interview I did recently with her, where I asked her about her experience living with RP. If you'd like to see the full interview, you can find it right down below in the description. Now, some of you might have remembered when I said that it's a genetic disorder and started putting two and two together. And yes, I may have it myself. With the specific type of RP my mother has, I ended up with a 50% chance of having it. And while you can get genetic testing to find out early, I opted to leave it a coin toss. I don't know exactly why, but I just want to find out on my own. So far everything seems completely fine, but I'm not out of the woods yet in terms of the age that people usually start seeing symptoms. But I am almost there, so fingers crossed. Well, that's all I have for you today. If you'd like to learn more about RP, I'll include some further reading in the description. And if you want to help advance research revolving around blinding disorders, you can check out this QR code or the link in the description, which will take you to the donation page to the Foundation Fighting Blindness. As always, sources are down below, and if you have any questions, leave them in the comments and I'll try my best to answer them. If you'd like to see more psychology and neuroscience content, consider subscribing. If you liked the video, like it. If you didn't, dislike it. And take care.